evening, good whatever time it is with wherever you're tuning in from. Thanks very much for attending our May monthly education session. We have Shannon Mumenthaler, who is the Chief Translational Research Officer at the Ellison Institute for Technology. I was learning about that, and she'll give us a little bit more background on that. She's also an Associate Professor of Medicine at USC. And so she's going to talk with us about what they're doing associated with functional precision medicine. I'm excited to hear what she has to say. So Shannon, I'm going to stop talking and turn it over to you.
Great. Thank you so much, Bruce. It's wonderful to be here this morning. Hopefully everyone can see my slides. Let me pull those up. Oh, make sure. Apologies. Does that look okay, Erin? Okay, great. So again, thank you so much for the invitation to be here. I think that what I'm hoping to get from today's talk and feedback from all of you that are online is, some of the work that we're really thinking about, these kind of human relevant complex in vitro models. How can we use those in precision medicine? But in order to kind of get to that point, there's been a lot of work in terms of model development and also assay development. And so I really want to talk about how we're thinking of integrating these quantitative experimental models and computational approaches, and then leveraging that to kind of better understand cancer system as a whole, and how kind of under various different conditions and over time, can we gain useful information to allow us to have these kind of tailored interventions. And so I think that this is actually, well, 2025 has been quite a tumultuous year, I know, for a lot of us in science. I think this is also very timely in terms of this particular society and the work that's being done here, given really the doubling down and emphasis from FDA and NIH on these human relevant models, and both kind of the goal of them being utilized in drug development, but then also thinking about them in terms of biomarker space as well. So with that, here's my disclosures. So I don't need to spend a lot of time kind of motivating this work. We know that specifically in cancer, we have a lot of work still to do in terms of success rate getting drugs into the clinic. So we have a poor kind of overall success rate, less than 5% for oncology drugs in particular. So how do we improve methods to address those critical gaps in metabolism, toxicity, and in particular efficacy, and thinking about also not only drugs in development, but once they are in clinical use, really which patients should be getting those drugs and when. And so I think there's been this, paradigm shift in terms of biomarker development, and it's really offered kind of more effective and personalized treatments that are tailored to each patient's individual biology. But again, we know that there's really challenges, and those really come at this tumor heterogeneity aspect. And so really knowing that molecular profiles are not always predictive due to complexity of signaling pathways, the tumor microenvironment, and just biology that we don't really understand. And so I think this is where there's the opportunity of these functional ex vivo models, and they're really poised to play an important role in precision oncology. And so our approach at the Institute, and this is work really that we've been thinking about for a number of years, is how do we start to integrate multi-scale context-specific data for functional drug testing? And we really see kind of the opportunities to build out these models, combine the models with various different multi-omics data, utilize AI and machine learning to really analyze kind of this large amount of data and incorporate that as well with some of the clinical information. Right now, I will say a caveat in what we'll discuss is really more on the left-hand side, because of building out the models and the workflows. We're using this more in kind of that preclinical or discovery research space, and developing this living biobank for identifying, novel drug candidates, or really assessing drug safety and efficacy. Obviously, the goal for a lot of us is to get to kind of the right side, which is this model-guided clinical decision-making. Can we use them to really predict patient response? And so hopefully, and I know there's others that are part of the society that are really doing some great work there, and that's our intent as well to get there. And so just to kind of go through a little bit of an outline for today's talk, I'm really going to cover two different parts. The first is really focusing on the organoid workflow, and I think highlighting, again, I know there's a lot of groups that are doing organoid research and really looking at it from a drug screening standpoint. One of the challenges is really around the assays and the data that we collect from these organoids. And so I really want to walk through kind of how we're leveraging imaging and AI image analysis to really get more of the temporal information and understand really the organoid level heterogeneity aspect. And then kind of I'll go into the second part where we're really bridging multiple complex in vitro models, so really organoid with organ on a chip, and hopefully honing in on kind of the rationale for that and why we need kind of this increased microenvironmental complexity, and really highlight a vignette of some of the patient-specific information that we were able to pull out from this colon cancer on a chip model. So for the first part, this is really kind of the traditional workflow that a lot of groups are doing, depending on the various different cancer types that they're focused on, but really thinking about patient-derived organoid-based individualized drug response testing. And so it's really getting these primary or metastatic tumors, generating the organoids, and then doing kind of a higher throughput screen, looking at various different drugs and having a number of different readouts. Now, one of the big kind of pushes has been initially a lot of the readouts have been more of a traditional cell viability assay. So kind of looking more at or not really having the heterogeneity aspect being captured, but really more a single time point, single readout, and kind of lumping all of the information of all of the organoids from a given patient as one number. I think then there's been more rollout of imaging workflows and other things really to kind of dive into other characteristics of these organoids in response to drug treatment, such as size changes, other kind of readouts of viability, and then bringing in some of the complexities with other microenvironmental cell types, other stromal cells, and various cytokine releases and other features. But still, a lot of the work that's being done is kind of a single modality, a single readout, and generally not including the temporal dynamics. And so one of the arguments that we want to make is really being able to dive into those dynamics to really understand both the intra and interpatient heterogeneity with these organoids, especially as we start to think about some of the new drug modalities that are coming out and how important it is to look at those dynamics. And so here's kind of an overview of the methodology that we're doing, and we term this SCOPE, so systematic classification of organoids for phenotypic evaluation. And so really what our workflow is, and we've been primarily piloting this in colon cancer, but we are also moving into other solid tumors and generating kind of organoid biobanks for colon and breast and others as well. But the work that I'm going to focus on today is just really in colon. And so we built out kind of our organoid biobank, again, primary and metastatic. And then for our drug testing or drug response workflow, we seed our organoids in multi-well plates. We're using kind of an automated liquid handler to do this. And then initially we looked at the FDA-approved oncology drugs. So we wanted to kind of take known drugs and look at the response across a number of different organoid lines. And so we did this at a single concentration. Well, actually, we did a couple different concentrations, but of these 166 oncology drugs. And then we used our imaging workflow. So we captured bright field images of organoids. So this is really a label-free imaging workflow over time. And then we utilize kind of either the traditional endpoint imaging as well as viability. So the cell titer glow, which is kind of one of the main assays for organoid viability. But within this imaging workflow, what we then leveraged was a neural network image analysis that allowed us to get both kind of organoid viability. So we could look at live and dead changes in organoids, individual organoids over time per patient, as well as because we were tracking these organoids over time, we used mathematical modeling to really look at organoid growth dynamics. And so we're kind of using this multimodal screening and phenotypic classification of drug response by combining these various different metrics of organoid phenotype. And please, if there's questions that come up throughout, I'm happy to take them. Otherwise, I'm also open at the end as well. So just to give you a little bit of background of our image analysis workflow, and so in terms of the training, how we set this up, and so we have our organoids. Initially, we used GFP labeled organoids just to help with kind of segmentation of the individual organoids, but everything subsequent to that was label free. And then we input those raw images. We do a kind of 2D projection, and then we're able to get segmentation of the organoids. We then have specific kind of training labels. So we have our control media, as well as our dead organoids, where we've introduced staurosporin to kind of really, kill the organoids. And so that was our way of being able to label live and dead organoids and pull out kind of morphology and size features and other aspects. And then that went into kind of the neural network training. And then in terms of processing, so again, this is done in a high throughput manner, multi-well plates, multiple organoids, multiple drugs. We combine our time series data from the imaging standpoint. We then do the neural network processing, classifying live and dead. We then track the individual organoids again over this period of time, because they're also in an extracellular matrix. They're not really moving much, so it's easy enough for us to track. We can also take them out much longer if we want. And then from there, we do our organoid growth modeling and viability. So here's just showing kind of a image over time of the organoids treating with a specific drug. And you can see the labels are the neural network classification of live and dead. So live in blue, dead in red. And then you can see, hopefully, those little tracks are actually showing how we can keep the information of each individual organoid. So we're pulling out all of that information within a given field, and then within, obviously, across the entire plate. So one of the first things we did before we went into kind of the drug testing arena, we realized we needed to just really understand organoid characteristics and growth properties of the organoids, because that's going to influence drug response classification. And also, we just generally didn't have a good understanding of the growth dynamics of individual organoids in these in vitro cultures. And so here, what we did is we took our organoids and a number of different patient-derived organoid lines. We did the same workflow as I mentioned, where we track the organoids, we then get the size and morphology information, and then we basically evaluated a number of different growth models, unconstrained and constrained, and then we used Bayesian information criteria to determine which model actually best fit the time series data. So you can see here, just again, looking at 3 different organoids as examples, you have very different kind of characteristics in terms of growth across those. And what we found was that the organoid growth was actually well described by the Gompertz model, which is consistent with others who have really looked at kind of tumor growth models in vivo, and Gompertz model has been kind of the more relevant model to use. And so here's just showing, again, for these 3 different patient-derived organoids, you can see both the inter-patient heterogeneity, as well as what we're showing is kind of the log 10 of the growth rate differences of each individual organoid within a patient. And so it's really the intra-patient organoid growth variation. And so I think this is really important as we're thinking about drug resistance and other features where you're wanting to look at kind of the underlying heterogeneity within a tumor itself, a tumor population. And so, here we can actually get that individual readout of each organoid for a given patient versus doing some of the more traditional readouts where you're collecting kind of more of a homogeneous readout of all the organoids and assuming they're all similar. You can see there's quite a wide distribution of organoid growth rates within a given patient. So the other thing that we wanted to do, so that was kind of the initial work, was just looking at organoid growth modeling. And then when we moved into more of our organoid drug screen data, looking at, again, these FDA approved oncology drugs and seeing response within the organoids, we first wanted to just confirm our imaging method. So this imaging time course and doing really neural network classification of live and dead, we wanted to compare that back at a single time point. So our neural network at day 5 to the cell titer glow, cell viability at day 5, just to see, in general, are we getting similar classifications of viability using our imaging classification. And here we're just showing, this is, the 166 different drugs for a given organoid line. And we're plotting the Z factor and showing when we actually correlate kind of the neural network percent to the cell viability, that we get a fairly strong correlation between those two. And here's just zooming in on some of the known kind of chemotherapy agents that are used in colon cancer. And again, you can see pretty consistent responses with cell titer glow at day 5, and then our neural network image analysis at day 5. But why do we need to do kind of the image tracking, the organoid tracking, and collect kind of organoid growth and viability? Hopefully, this is kind of to try to highlight some of that. So when we zoom in on some other drugs within this panel of oncology, FDA-approved oncology drugs, we again see by the Z factor, if we're looking at the cell titer glow and our neural network, they look very similar. So this is kind of traditional method, limited information, so single time point, and really just kind of collecting all of the organoid information as a single readout. When we then go and look at the multi-time tracked organoid size, you again see for these two different drugs that the actual organoid area looks very similar. Basically, it's a flat line. They're not growing in the presence of these drugs. So you might interpret them as having a similar effect on this particular organoid. But then when we also combined, again, the growth rate information with our neural network, with the live dead classification, this is when you actually start to see distinctions. So using our specific kind of classification workflow, you now can see that in the osimertinib treatment, you're getting really this change in viability. And so it's really causing organoid death within over the period of time that we're looking, where the other drug is really showing that the organoids are still alive, they just didn't grow. And so we can start to really distinguish between more of a cytotoxic versus cytostatic effect, that again, if you only had a single readout, you wouldn't be able to determine that. You would kind of assume that they were having the same effect on the patient organoids. And so then what we did is we took this, that's just kind of showing one example, but what we can do is look at it more holistically. And so we call this kind of the 3D growth viability plot, or GV plot. And so this is combining modeling-based growth score with our neural network image-based viability. And so now we can look at all of the drugs in a 3D plot, really showing the growth score, and then over time, the changes in viability. And so what we find is that when we look at our DMSO treatment, that's kind of up here in the upper right-hand corner of this 3D plot, where again, the growth score is close to 1, and most of the organoids are viable over that entire course of the experiment. And then in the other extreme, down here in the bottom left-hand side, we have our staurosporin treatment, which again is our control for inducing organoid death or cell death in general. And you see that here, you have a very low growth score and low viability. And then when we overlay the other 5-FU, 5-fluorouracil, and SN38, which is the active metabolite of irinotecan, again, common chemotherapy agents used in colon cancer, you can see they fall kind of in between those two extremes, with 5FU being more kind of cytostatic. So, there is an effect on the growth score being lower, but in terms of viability, the organoids are pretty viable, versus SN38, which is closer to the staurosporin, reduced growth score, reduced viability. So then when we overlay that now, looking at kind of other drugs maybe of interest and really start to look at the quadrants within this 3D plot, you can start to see phenotypic classifications. And we also went through, this is again using our imaging workflow, but we also did some manual classification of this as well, just to confirm that our neural network classification was accurate. But you can start to really quadrant off the regions within the 3D plot and show that the drugs in this area are more cytotoxic. You have then your more cytostatic drugs. And then here in the middle, you're getting some interesting time dynamics that we can pull out. So some where you're getting more late cytotoxic. So they seem to be growing. Here's an example where the organoid between time point 0 and 3 look similar to DMSO, but then all of a sudden within those last kind of two days, you start to see a change in organoid area, and then also a real difference in terms of organoid viability. So you get this large drop off in percent live organoids. And again, we can really do spot checks across all of those and see the differences between the different phenotypic classifications. And so that's doing kind of the earlier work if you wanted to do more screening across a number of different drugs, maybe a single concentration and a number of different organoids. Then from there, if you want to select specific drugs of interest based off of where they fall within the classification plot, we can then use the same kind of workflow to capture dose-dependent effects across those different organoids. And so in this case, we picked 2 different drugs within the FDA-approved oncology list that target EGFR in different ways. And we looked at 6 different patient organoids for gefitinib and then 6 for osimertinib treatment. And here we're plotting the IC50 for viability, again, using our neural network, as well as IC50 for growth, using our growth score. And you can start to see some of for some patients, there really is no difference between viability and growth. So you really get kind of an overlay in terms of what's happening from a response. And then other ones, you get more of this initial dip in cytostatic effect at a lower concentration, and then the cytotoxic effects start to come at later, higher dose ranges. And so, when you're starting to think about for given organoids and for given drugs, you could start to really hone in on specific concentrations of interest depending on the effect you're looking for. And ultimately, we can then start to look at the comparisons and kind of the similarity or differences across different organoids, lines across different drugs, and really see the effect on viability and growth. And so one of the things that we actually looked at, again, this is a small sample size of organoids. We are actively kind of expanding this out, but we have 3 of our organoids that are KRAS wild type, 3 that are KRAS mutant. They also have a number of other kind of mutations and in varying stages and other factors. But we could just look at kind of how similar they are in terms of their growth and viability response to 2 different EGFR targeted agents. And one of the things that we noticed is that, yes, in general, the KRAS mutants kind of cluster together and the KRAS wild types also do, but there are differences. So there are some that are less responsive to specific drugs like osimertinib, or you have up here with gefitinib, even though these are your kind of 3 KRAS mutants, you do see that there's more of an effect of this one against gefitinib. So this is kind of highlighting that while molecular markers may help stratify in some cases and for some drugs, really being able to do kind of the functional testing and looking at phenotypic classification, both viability and growth, really gives you additional insights into the responsiveness and kind of how you may think about even combination therapies or dosing regimens. And so, this is kind of the initial workflow and just trying to show some of the additional advantages that we or additional information that we think you can glean from this approach with the imaging and getting the time dynamics, but open to other suggestions and thoughts for this. And again, this is all kind of done in an automated fashion. So I just wanted to kind of take a minute just to summarize this first part and some of the improvements that are ongoing. So again, how we see using this classification of organoids for phenotypic evaluation is really combining kind of the dynamic or time course imaging with AI image analysis and tracking. So you get individual organoid growth dynamics as well as viability classification. We can then use this kind of 3D vector to look at where the various classes of drugs and by organoid, so by line, and really look at the effectiveness in those specific patients, and then start to subtype based off of functional response. So at this point, we're really working right now. The neural network that we've been using mainly has been really kind of proprietary. So we're looking to expand to publicly available neural networks. We're obviously increasing our organoid numbers and cancer types. We know that this is a small subset. And then I think the other important point and work that is ongoing is introducing some of the other aspects of the microenvironment and ensuring that we can do a similar kind of workflow in terms of segmentation and classification. So I will then kind of switch gear, not really switch gears, but kind of take you through another workflow that we've really been spending a number of years establishing, which is our organoid on a chip development. And so kind of the motivation behind
Shannon,
this.
can I interrupt before you proceed?
Yes.
Hi, this is Constantine. Sorry for interrupting the flow, but I hope it's a good time to
Oh, this is a
ask
perfect
a question,
time.
relevant to the first part. I mean, this is very important work that you're doing in terms of dissecting the cytostatic versus cytotoxic effect of drugs in these organoids. I suppose one advantage of the organoids is that they have not been cultured for decades in plastic. They're not maximally growing. And a key argument has been that that by itself can help the organoids give a more accurate assessment of or prediction or extrapolation of clinical activity. Have you had a chance to, I guess, adapt some of those organoids to the culture, see if they proliferate fast, and apply the same treatments and be able to show that there is a higher fraction of these 2D culture adapted organoids in which you see the separation between the curves, the cytostatic versus the cytotoxic assessment? And I'm asking this because if you have those data, that can be a very powerful support for the idea of using the organoids for those types of assessment as opposed to the adapted organoids, because there's a lot of discussion in the field about, there's a lot of great stuff we can do with the organoids, but it wouldn't be great if we can adapt them to the culture so that we can do this type of screen or that type of screen. What are your thoughts on this?
Oh, interesting. Yeah, no, that's a really important point. We have not, right, we've gone kind of the more 3D workflows and really kind of moving it that way versus kind of trying to get back to a 2D system and compare. We've had trouble, at least with our organoid, the primary cultures, if we have taken them out of kind of the extracellular matrix and tried to seed them on, they don't really thrive well in kind of plastic cultures. And so again, one of the reasons why we've just kind of stuck with the 3D. But that's an interesting point, and I get your kind of reasoning for asking that, and I mean, it's important. We do a lot of work to do this in 3D, and it's a lot of kind of from both the wet lab experimental work as well as the imaging and image analysis. Like it's cumbersome, and so is it worth all of that. I think that is the big question that I still have of like, you know, are we able to really make useful insights with this workflow compared to just a 2D kind of screen or more traditional?
And to be clear, Shannon, I completely understand and have experienced the challenge of trying to take a material that can only grow in 3D and take it into 2D. Right? I agree with you that that's an entirely different area that takes huge effort and time, resources, etc. So I don't advocate that this is done routinely unless someone really wants to do it. I'm only asking from the perspective of what additional data are there, or what aspects of the data can, from what you've done, can give more confidence, I think, to the field that, hey, you know, you should be using the 3D organoids to assess drug response, because you're not going to see as frequently the separation between the cytotoxic and cytostatic effect.
Yeah.
And I guess I missed in your presentation, but overall, from the organoids that you've examined, like how frequently do you see that there isn't a separation between the cytotoxic and the cytostatic, or there is a major separation between the cytotoxic and the cytostatic pattern of response? And how does it perhaps compare with your expectations based on historical data in 2D?
Yeah, no, that's a good point. I mean, again, it's a fairly small number at this point, so I don't want to make huge claims, but I think in terms of being able to, I think where we're seeing interesting utility is the fact that if you just do a viability assay readout at the end, you don't really know, right, how much of that is a cytotoxic versus cytostatic effect. If you just look at organoid size, you also don't know, again, from just an organoid growth difference, what's happening. And so I think it's the combined information that's getting us the stratification. And so I think that's where, you know, going back to even in the beginning when I showed the correlation between cell viability and neural network at the end, I mean, there's a strong correlation. It's just the interpretation then now, once you build in all of this other information on the time dynamics, you would have a different outcome. You would think that it's a, you know, whatever your rationale is for that drug with the cell viability, that it's killing them, but in reality, it may just be slowing growth. And then obviously from a standpoint of resistance and other things, that is a critical piece of information that you would want to have in terms of drug selection. So, you know, we do see clustering by the different organoids, by different drugs. And so we can kind of separate out certain organoids have a similar response to a class of drugs versus others. So we have that information. But, you know, I don't think we can make any claims about historical data or any of the 2D at this point. That's good food for thought that I need to
Thank you.
add a little bit more.
Thank you for, sorry for the long question, but thank
No,
you
I think it's
for the
really,
clarification and great initiative, great efforts there.
thank you. No, I think it's
Appreciate
very important. I appreciate that. Any other questions on this part? Okay. And I guess one of the other things I didn't talk about, that a lot of this data also, we work closely, as I mentioned, with mathematical modelers, so some of the organoid growth, but also we work with groups that do agent-based modeling. So again, being able to get a lot of this quantitative information from these organoids and then parameterize these agent-based models where you can simulate kind of 3D tumor growth and then do in silico perturbations is really where a lot of the genesis of this work came from, and some of the work that we're continuing to do. And so I think that's also it within the time window that we have, we can do this classification of cytotoxic, cytostatic, or even some of the late effects. But being able to incorporate more in silico based approaches and look at longer time dynamics, and then be able to really look at emergent phenotypes and really predict on the longer time scale is really where a lot of this is headed to. So just wanted to put that out there. Okay, so for the second part, as I mentioned in the beginning, one of the things that we wanted to do was think about other aspects of the microenvironment. And so while organoids, especially for kind of this drug screening workflow and thinking about them in terms of biomarkers for individual patients and being used to look at treatment response, they are missing certain aspects of the milieu or the ecosystem. And so we really wanted to combine organoids with organ on a chip. And so we started this work a number of years ago, where a lot of the organ on a chip field was coming out more from a toxicity space and really developing normal organ chips, looking at organ level physiology and functionality to look at toxicity assessments, and then starting to look at more at disease modeling. And so we then took that and wanted to adapt kind of an intestine on a chip model to a colon cancer on a chip model, to bring in the tissue-tissue interface, the biomechanical motion, so really being able to recreate the organ level biomechanics that are happening, the multiple cell types, and then obviously the fluid, because these are microfluidic. So really having fluid exchange, looking at shear stress from fluid flow, and the vasculature. And then obviously there's been a number of efforts really building even on these single organs into multi-organs. But what I'm going to talk about today is really the adaptation of kind of single normal organ into a cancer on a chip model and the work we've done there. And so really being able to kind of bring out this complexity of colon cancer biology. And so we know that there's, with many solid tumors, the kind of complexity of the microenvironment with the immune and stromal and endothelial cells. But in addition, really what's unique about colon are these shear stresses from the fluid flow. So as the tumors are growing into the lumen of the intestine, they're undergoing a lot of mechanical forces from fluid flow, as well as this cyclic stretching like motion that's occurring from peristalsis within the gut. And so here's just showing, we've generated a number of different iterations of this model. And so what we're doing is we're taking our colon tumor tissue, we're generating the organoids, banking those, doing a number of the studies I just showed you before, but then we're also taking these organoids and seeding them onto this microfluidic device, which again is, the chips are made of PDMS, they fit in the palm of your hand. Here's the cross section of the chip for those, we're working closely with Emulet and their model. And so here is, as I mentioned, a cross section. So we can see our epithelial cells in the top channel. It's separated by a porous membrane. And then in the bottom channel is where we seed our endothelial cells, kind of to line the bottom channel to form a tube-like structure to mimic a blood vessel. You have fluid. We can control the flow rate in the top and bottom channels. And then what's really unique about this particular organ on a chip is the vacuum chambers that line the length of the channels. And so this is where you can introduce the cyclic strain to really mimic peristalsis in the gut. And so now you have both the biochemical as well as biophysical or biomechanical forces recreated in this model. Our initial version of this was really based off of cell lines. And so we had kind of normal intestinal epithelial cells with our colon cancer tumor cell lines. And then lining with HUVEX in the bottom. Then we've really adapted this to a patient-specific organ on a chip model. And so we use our organoids, varying stages and other backgrounds, and we see those in the top channel. And then our endothelial cells are human intestinal microvascular endothelial cells. We've then layered on other cell types such as cancer-associated fibroblasts, so autologous cancer-associated fibroblasts, and then more recently have spent time really working on an immune competent model, and so really looking at immune cell recruitment with PBMCs flowing into the bottom. So I'll show a bit of kind of the model development, and then if we have time, one of the reasons why I think that this kind of patient-specific model is going to be so valuable in terms of really looking at therapeutic response. So here's just showing kind of going through the bottom. That was the endothelial cells. Now we're moving up through the porous membrane into the top channel, our normal epithelial cells, and then our tumor cells in green. And so you can see again these nice 3D structures that are formed within the chip, and also the fact that you have this tissue-tissue interface, so you can really look at epithelial to endothelial interactions. We can do a number of different readouts, looking at cell phenotyping, invasion and metastasis using imaging, traditional viability readouts, and then because of the microfluidic aspects of the model, we can look at effluent analysis. So we're continuously collecting the effluent and doing various different multi-omics, in particular looking at metabolite changes over time in a non-destructive manner. And again, there's a number of applications that we can look at. I think ultimately, especially for the relevance of this group, is more on kind of functional drug response and ultimately identifying novel targets. This is just the patient characteristics of our organoids that we generated. And so then you can see pre-digestion organoids grown in the traditional dome. And then when we kind of digest those and then seed them on the chip, they form various different kind of morphology and structures dependent on each individual patient. And then the endothelial cells are seated in the bottom channel. Under flow, we can do RNA-seq, really looking at kind of differential gene expression, showing patient level heterogeneity. And then ultimately we compared the gene expression of 3 different models. So we have our tumor tissue, and then we have the organoids generated from that tumor tissue, and then we have the organoids on chip. So kind of building up the complexity and seeing how similar in terms of the transcriptomic profiles the organoids and the organ on a chip are back to the matched tumor tissue. And what we found is that our organ on chip or organoids on chip were actually closer to the tumor than the organoid only. So again, we're not perfectly mimicking the tumor. We know that there's a number of things that are missing, but in terms of kind of transcriptomic similarity, we're getting closer to the tumor when you build out more of the microenvironmental complexity in these models. Again, we can do a number of different characterizations, looking at kind of tight junctions in both the tumor, the epithelial channel as well as the endothelial channel. And then we do barrier function analysis, really looking at, can it form an intact barrier and how long does that take by each patient. And so again, by the time we start our experiments, we have a nice intact barrier formed on chip. We can then do readouts such as invasion and viability. So here we're looking at using imaging. So this is a cross section looking down the chip. Again, in red are our endothelial cells forming a blood vessel. So the empty black space here is actually where fluid flow is going through, separating. You have the porous membrane, and in the top channel you have your tumor cells. In this case, we labeled them with GFP. So at the start of our experiments, we have that nice separation between the epithelial and endothelial channel, but over time, the tumor cells will start to invade through the extracellular matrix and into the bottom channel and either adhere to the endothelial cells or flow out into the reservoir. And so we can actually quantitate this invasion ratio. We can also do metabolic imaging using fluorescence lifetime imaging. So these are all kind of non-destructive ways of interrogating the biology and looking at different, in this case, metabolic shifts between our different patient organoids on chip. I'm going fast, I know, but I think we're running out of time. And so then again, we can also do cell viability. This is all done on the same chips. So we can collect the effluent, and one of the things we first look at is just if we're introducing drugs, we want to ensure that there's no absorption of the drugs onto the PDMS chip, and so we can use that by look at by mass spec and see that in this case for the drugs that we're interested in at this point that there's no absorption over time. And then when we look at viability, and this is looking at 5-fluorouracil, again, a drug given in colon cancer. We have our organoids that are pre-treated, and then organoids from patients that were treatment naive, and we see actually a separation, so suggesting that the pre-treated patient organoids are more resistant to 5-fluorouracil than the treatment naive. So this is just generally giving you a sense of the data that we can collect on these chips, and now we're really expanding the patients that we can build out, and then looking at the various different drugs of interest. I did just want to highlight like I said, some of the complexity, especially depending on drug modalities of interest, important to think about the stromal components. And so here we can really layer in cancer-associated fibroblasts that are isolated from the same tumors that the organoids are. And so we can layer them onto the chip and really form the sandwich. So now again, you have your endothelial cells in the bottom, your porous membrane, you have your cancer-associated fibroblasts sandwiched by two different extracellular matrices, and then the matched organoid seating on top. And we can look at things like invasion or drug response with this increased complexity. And then now we're also looking, as I mentioned, with immune cell recruitment. So here our endothelial cells are there. They're just unlabeled, so you can't see them, but we're flowing in PBMCs into the bottom channel. So you're getting attachment of the PBMCs to the endothelial cells. And then you're getting this kind of immune cell recruitment and extravasation of the immune cells into the epithelial channel, as well as you have your tumor cells that are invading into the bottom channel. So we can measure both kind of extravasation of immune and then intravasation of tumor cells and any viability metrics and cytokine changes. So specifically looking at some of the antibody therapies in cancer, this kind of building out more of an immune competent model is going to be important. And so, you know, we've looked at kind of HLA matched versus not matched donor, and then also thinking about autologous PBMCs as well. I don't know if there's questions on this part. I'm happy to, I had a few more slides to go into this specific vignette, but I also am cognizant we only have 8 more minutes. So maybe I'll pause and see if there's specific questions. I wanted to just kind of highlight the model development and all the capabilities around a patient-specific organoid on chip and the interactions with the kind of microenvironment and the capabilities of looking at patient-level response. There's no questions there, then I will maybe take a couple minutes just to highlight one of the things that was really informative as we started to develop this model, and we kind of stumbled across more of the interesting findings within colon cancer biology and this role of neurotransmitters. And so this really came about by working with a medical oncologist who was doing a lot of interrogation of this large randomized phase 3 clinical trial for metastatic colon cancer patients. And in particular, there's been some interest in neurotransmitter involvement in colon cancer, and really this dysregulation of neurotransmitters and the involvement in how that changes intestinal function and homeostasis. And so when he was looking at a lot of different neurotransmitters and really looking at biomarkers kind of worse overall survival and progression-free survival, it actually came about that high GAD1, which is involved in basically synthesizing GABA, as well as low ABAT, which is involved in catabolizing GABA, were associated with worse progression-free survival and overall survival in these metastatic colon cancer patients. So it was really suggesting that kind of having high levels of GABA had an overall worse outcome. And so we wanted to be able to dig into that a little bit more. There's been some work that's implicated GABA in colon cancer, but it's kind of had conflicting evidence between in vitro and in vivo. There's been a lot of work looking at it more GABA involvement in kind of CNS metastasis. And so we wanted to utilize our organ on a chip to better understand that. And where this led was looking specifically at kind of knocking down GAD1 initially in cell lines and showing that we could reduce invasion both by knocking it down as well as pharmacologically inhibiting it with 3-MPA. We also showed that if you exogenously added GABA, you saw this increase in invasion. But where the patient-specific information and really this kind of developing these personalized or functional models came into play was when we started to dig into GABA across our different tumors that we had. And we started to notice that GABA levels there was a clustering where you saw high GABA intensity in the KRAS or RAS altered tumors compared to the RAS wild type. And so that was something that we wanted to be able to really dive further in. And this is where going back to our organ on a chip model and really having a functional model to be able to look at the behavior of these cells on chip. And so what we found again is when we looked at our RAS altered organ on a chip models, we saw higher GABA levels in the RAS altered versus the RAS wild type, both in terms of effluent, so measuring GABA in by mass spec based metabolomics, and then also by IF. Again, these are our RAS altered organoids on chip, and we see higher GABA levels. We also, again, see high GAD1 expression in the RAS mutant. And so this was something that, again, we wouldn't have been able to really uncover this kind of stratification based off of molecular alterations without having kind of these patient-specific models to be able to do some of this testing. So we went and did more treatment kind of to knock down or to inhibit GAD1, so really reducing the level of GABA on chip. And again, we see the effect only in the RAS mutant or RAS altered chips where you see this reduction in invasion. You don't see that in the wild type. And then we did isogenic cell lines as well, just to confirm the same effect, and looked at also EMT markers as well. And then we went back to TCGA and really then looked at the stratification and saw that in the RAS mutant colon cancer patient samples, you see high GAD1, and then again, high GAD1 leads to overall worse outcome in these patients. So that's just kind of summarizing the second part of the talk, is just how you could take kind of this clinical observation with this clinical trial data showing this link between GABA and kind of the levels of neurotransmitters and association with patient outcome, and then using these patient-specific models to really look at the behavior of the tumor cells and showing that potentially targeting GABA synthesis in RAS-altered colon cancer cells could have a strong outcome compared to your RAS wild-type counterparts. And so with that, I am happy to take any questions. Just a huge thank you to patients and their families, also the collaborative team, both at the Ellison Institute as well as our collaborators at USC and University of Minnesota, especially on the modeling side with Jasmine Fu and Einar Gunnarsson. And the funding, this was supported by a tissue engineering collaborative as well as a UO1 in the Cancer Systems Biology Consortia. And then I'm happy to take questions.
Yeah, Shannon, Bruce, thanks very much. I mean, just a lot of been very interesting information. Hey, question mark for you, the first part of your talk. You were looking at individual drug responses. How do you think about drug combinations?
Yeah, no, I think that's an important point, and actually something that we've been discussing on, especially from a modeling standpoint. I think we can start to look at those iterations, but I think that's exactly where having this information about kind of the cytotoxic versus cytostatic response and how you might want to pair those together is going to be informative for combination. So we haven't gotten there yet, but I think that's one direction that you could really take this and utilize and think about more informed strategy design, understanding kind of the responsiveness to individual organoids.
Does anybody else that's online have any other questions or comments? Shannon, if people want to reach, how can they reach you?
Yeah, so you can definitely send me an email. I'm part of the Ellison Medical Institute. I did just want to give kind of, this is our institute. We're in Los Angeles, and the goal really is to have kind of the infrastructure that really supports our clinic, which is actually up here on the 5th floor, and then we have our research labs on the 3rd and 4th. So it's really this multidisciplinary interaction and patient-centric research. And so, love to have anyone come and visit us in Los Angeles. And, this is really, we're trying to leverage technologies to really better understand disease and and well-being. We have the clinic that really runs from prevention all the way to cancer as well. And so these
All
are
right. Yeah, thanks for sharing what you shared with us today. And for anybody who's on the line, we're still looking to fill our speaker lineup for 2026. If you or somebody that you know has some interesting work you think you'd like to share with the group, please reach out to someone at SFPM to be able to share that. Our schedule right now is open. The last thing to look at is, look, we're always making sure that if you're not a current member, please join. Right, these are one of the things we do on a monthly basis in order to be able to have this spread the news associated with this area of FPM, which we think is exciting, obviously, not only for just research, but also down to patient management activities. And the last thing is, if you know somebody that you think you should drag in here, refer them in here and let's get them, part of this group so we can work on helping inform, spread education, and get people to advocate further for this technology platform that I think is important, obviously, for research and for patient management activities. Shannon, thanks again. Appreciate your time today.
Thank you so much.
Have a great day.
Thank you.
All right, thank you. Bye-bye.